Hello, my name is Chiebuka Okpara. I'm a senior biomedical engineering student at the College of New Jersey. I'll be discussing the research I did as a New Jersey Space Grant Consortium intern. NASA and SpaceX have plans to return to the moon and travel to Mars by the end of the decade. When the astronauts travel into space, they are no longer protected by the Earth's magnetosphere and are exposed to galactic particle radiation and solar particle effects. Long-term exposure to space radiation induces high linear energy transfer, causing oxidative damages to bone cell structures, thus decreasing the stiffness, thickness, and elastic modulus of bone, which can lead to bone fractures. For this preliminary study, the objective is to evaluate the effectiveness of the computational micromechanical modeling in determining the bone elastic modulus for different radiation treatments. All live animal work was performed with our collaborator at Johns Hopkins University. Rats were irradiated at Brookhaven National Labs and were exposed to 100 centigrade proton radiation at the tibia head only. This study only has two samples, one for the control and the other for the irradiated. Micro CT scans were taken at 2-micron resolution. In mimics, micro CT scans were imported to generate a 3D representative volume element, or RVE. For each specimen, 10 anatomical regions of the cortical bone were analyzed. Bones were segmented from the pores at a set pixel intensity threshold of 121 gray value. A custom image processing code was written for MATLAB to convert the RVE into micromechanical models. The code will recognize the DICOM files, downsample it into a new volume, and assign threshold values for bone and air. MATLAB's exported file was then imported into the MIcGMC software, which stands for the Micromechanics Analyzed Code for Generalized Method of Cells. The code was provided by NASA Glenn Research Center. It was traditionally used to calculate the mechanical properties of composites used in aerospace, but it can be adapted for biological tissues. The software ran numerical simulations to determine the effective elastic properties of composites by assigning material properties to each subcell. Traditionally, finite element analysis has been used to define structural integrity, but GMC is better at computing the material properties of bone since it relies on details of the microstructure, while finite element relies on the overall geometry. Currently, the MAC file generation code still needs refining, but the porosity of the bone samples was determined. R was used for statistical analysis, and a two-way ANOVA was used to compare the radiation treatment and downsized volume to the porosity. A post-hoc Tukey test, an alpha of 0.05, was performed to account for each individual treatment comparison. Figure 2 shows that there's no significant difference between the RVE downsampling factors. The post-hoc results revealed a significant difference between the radiation treatments and porosity. And Table 1 shows the numerical values of porosity for each treatment at all volume levels. This is the first study to investigate the effects of radiation on the material properties of bone through micromechanical modeling. The results support a previous microindentation study which suggests that bone stiffness decreases with greater exposure to alpha radiation. This study shows a trend that irradiated groups correlate to significantly reduced elastic moduli. The large variance is due to the variability of the tibia's unique microarchitecture, partial volume effects, small sample size, and RVE index detail reduction. The bone's microarchitecture displays unique patterns of interstitial pores. During the RVE generation in mimics, it's imperative that the volume must be within the cortical regions of the tibia and avoid the empty spaces of the micro CT file or risk approximating the composite effective modulus to be only air. Table 1 displays porosity's downward trend as downsampling index increases. As the RVE size decreases, the detail of the model becomes more simplified. This runs the risk of being non-representative of the bone since there's less computational subcells to compute the effective modulus. Future work consists of finalizing and analyzing the RVEs using the MIcGMC code. And to conclude, I would like to thank the New Jersey Space Grant Consortium for funding this work throughout my academic year.